Hello, good morning and welcome to all of you. I am Rajesh Kumar Singhal and I am here to present a new lecture on the long distance transport of the nutrients. So friends, in the last class we have discussed about the short distance transport of the nutrients. And in this class we will discuss about the details of the long distance transport. So friends, you know that the nutrients are moved from the soil to the leaf, tissue or the cells. So soil is the source of the nutrients and the cells are the ultimate targets of the nutrients where they reached. Okay. So the first nutrient reach from the soil to the xylem. This is known as short distance transport pathway. Then nutrients transport from the xylem to the aerial portions or the cells. This is known as long distance transport pathway. Okay, so in the last class we have discussed that the nutrients first present in the form of ions in the soil solutions. Then they reach to the root and first they interact to the root cell walls where there is a formation of apoplasmic free space or the Donen free space. Then we have discussed that how the nutrients move from the cell wall to the epidermis, cortex, endodermis and the pericycles, and also discussed that in the endodermis there is a Casparian strips which block the apoplasmic transport pathway. So for this plant have symplastic way of the transport where the plasmodesmata have important role. And then at the end we have discussed that finally this nutrient reach to the xylem, that is known as xylem loading. So in this short distance transport of pathway at the two places, at the cell membrane and the xylem loading, at two places there is a involvement of transporters. Okay, so we already also discussed about the transporters, that there is some channels are there, okay, that is involved for the passive way of the transport. Then pumps are also there, active pumps. Then we also discuss about the carrier proteins. Okay, so there are three types of transporters which involve in the transport of nutrients at cell membrane and the xylem membrane. Okay, so this xylem loading is the last pathway of the short distance pathway and the first process of the long distance pathway. Okay, so very clear that this is the short distance pathway. Now in this lecture we will discuss about the xylem transport, that how the nutrients transport in the xylem. Then we will discuss about the xylem loading, that how the loading of nutrients take place in the xylem. Then we will discuss that exchange adsorption and the xylem retrieval, means how the nutrient also absorbed and the uptake back from in the xylem. Then we will discuss about the mechanism of release or secretion of nutrient from the other cells. Then we will discuss about the xylem unloading in the aerial parts. Okay, we will discuss about the phloem transport pathway. Okay, because nutrients are transported in both xylem and the phloem, but water is transport only in the xylem. Okay, then we will discuss about the retranslocation and cycling of the nutrients, means movement of retranslocation or the cycling of nutrients. Then we will discuss about the some factors which are crucial for the mobility of the nutrients in the phloem. Okay, we will discuss about the exchange between the xylem and phloem. Then we will discuss at final the remobilization of nutrient. That some case when the nutrient are deficient, plant remobilize the nutrient from the dead plant tissues or the other source tissues. Okay, so this is the pathway of the, or these all pathway include under the long distance transport of the nutrients. Okay, so friends, let's start the lecture. Okay, so the long distance transport of water, solute elements and the low molecular weight organic compounds take place in the vascular system of xylem and phloem. ओके, तो ऑब्जेक्टिव में भी पूछ सकता है कि जो न्यूट्रिएंट ट्रांसपोर्ट है which cells are involved, xylem and the phloem. Okay, these are the main transport pathway. Okay, or the basal elements, vascular systems. Okay, second point, long distance transport from root to the shoot occurs predominantly in the long living xylem vessels. Okay, so this is also a one of MCQ question, that which one of the following act as a predominant transport pathway, that which of the following involve in the predominantly in the long distance transport pathway. तो आंसर जाएगा आपका फर्स्ट थिंग, तो ये आपका नॉन लिविंग मींस ये आपका डेड टिश्यू है. ओके, एंड xylem vessels are involved, not tracheids. Okay, tracheids are not involved, vessels. Okay, then in annual plant species, long distance transport in xylem vessels may also interrupted by the tracheids at root and the shoot junction. Root and shoot junction, there is a tracheids which interrupted the long distance transport, or in the nodes. Okay, nodes of the stem. So this structure poses the internal resistance in the xylem volume flow, but simultaneously it permit the intensive xylem phloem solute transfer. Okay, so mainly at the internodes and the root shoot junction there will be a interchange between the xylem and phloem take place. Okay, fourth point is the xylem transport is driven by the gradient in hydrostatic, that is known as root pressure, and by the gradient in the water potential. तो जो आपका xylem transport है, the long distance transport है, that is driven by the two process. First is the gradient in the water potential, and second is the gradient in the root pressure. Okay, and fifth है the gradient in water potential between root and shoot is quite more or steep, particularly during the day when the stomata are open. So friends, what happen during the day when stomata is open, the transpiration rate is also high. So water potential difference between the root and the leaf are more. So gradient is more. So water flow or nutrient flow is also high. Okay. So मेरे हिसाब से all the points are very clear. Now come to the next slides. Okay. So this is the water gradient. It is high in atmosphere, then leaf cell, xylem cell, root cells, and the external solutions. Okay. Then next point is the solute flow in the xylem from the root to shoot is unidirectional. Okay, तो जो आपका xylem flow है that is the unidirectional. Okay, so आपका water transport है that is unidirectional, क्योंकि water आपका only xylem में flow होता है. However, under certain condition in the shoots, a counter flow of water in the xylem may also occurs. There is some exceptions, okay, like low transpiring leaves, okay. But in case of phloem long distance transport, the living sieve element tube cells are involved, okay. तो xylem में आपकी क्या थी? Dead cells, okay, basal cells. और phloem के लिए कौन इन्वॉल्व होता है? sieve tube cells, and this process is the y-directional. So nutrient transport of the nutrient is both unidirectional in the xylem and y-directional in the phloem. And both dead cells or non-living cells like xylem vessels and the living cells that is known as sieve tube cells, both are involved. So there is the difference between the water and nutrient is very clear. Okay, now come to the next point. The phloem transport is an important component in the cycling of nutrients. Okay, जो आपका phloem transport है वो क्या करता है? आपके nutrients का cycling that have important role in the nutrient cycling. Okay, between the shoot and the root. And for the signaling the nutrient status of the shoot to the roots. Okay, तो जो आपका signaling mechanism है nutrition deprivation और access के लिए, उसमें phloem transport और cycling of nutrient is important and for the signaling process. Okay, an element can enter the phloem in either to the shoots or to the roots. तो आपका देखा जो आपका root shoot junction होता है वहां से आपका एक तो हो सकता है, दूसरा आपका जब remobilization होता है प्रोसेस वो डिस्कस करेंगे. In this process there will be a transfer between the xylem and the phloem. Okay, तो root से भी हो सकता है और shoot portion में भी हो सकता है. Okay, during long distance transport elements and organic solutes are transported between the xylem and phloem by the extensive exchange process. Means during the long distance transport of the nutrient, exchange process are take place. Okay, so this slide is also very clear that how the transport in the xylem take place or long distance transport take place. Now come to the first point that is known as xylem transport. Okay, so the composition of the xylem sap depend upon the plant species. So these are the factors which affect the composition of the xylem sap. Okay, तो पहला फैक्टर कौन सा है? Plant species. Second, elements supplied to the roots. Means which elements are present in the soils and supplied to the roots. Okay, second point. Third point is the assimilation of nutrients in the roots. Okay, means types of nutrient transported in the roots. Or fourth point is the nutrient cycling which take place between the xylem and the phloem at the root shoot junctions. Okay, so these four factors affect the composition of xylem sap. Now the concentration of solutes, means concentration of solutes depend upon the first point is the dilution of water, means how much water is present. So if water is less, then concentration of nutrients or solutes are high. Okay, and second point is the transpiration rate. If transpiration rate is high, then concentration of nutrient is also high because water demand is also high. And third point is the time of day. You know that at the time of afternoon there is a high transpiration. Okay, so these are the factors which affect the composition of xylem sap and the concentration of solids. Clear? Next point. The eases the competition for the photosynthesis between the pores and root during the reproductive period, leading to the higher uptake and xylem loading of nutrients. Okay, so ये एक तरह से आपका डिमांड सप्लाई है, जितना आपका डिमांड होगा उतनी सप्लाई ज्यादा चाहिए. ओके, और मेनली आपको कौन से रिप्रोडक्टिव पीरियड्स में, बिकॉज दैट टाइम इट्स नीड फॉर द pore development and also for the root development in case of legumes. Okay, means in the legumes there is a competition for the photosynthesis, or you can say for the biennial or the perennial species. Both vegetative and reproductive phase comes together, so there is a competition for the photosynthesis, but not case in the cereal crop, because the reproductive phase comes generally after the vegetative phase. Okay, next, the proportion of various nitrogen fraction in the xylem sac also depend on the form of nitrogen supply. Okay, you know that nitrogen is a important nutrient for the plant growth and development, and the concentration of the or nitrogen fraction also depend in the xylem sac depend upon the form of nitrogen supply. Like nitrogen can be supplied in the form of nitrate, it can be formed in the ammonia or the nitrogen fixation product, or the predominant site of nitrate reduction is the root or the shoot, and the proportion of recycled nitrate from the sensitive tissue or senescence leaf tissue. Okay, so it depend upon the different form of nitrogen. Okay, also the concentration of organic acids in the xylem sac depend upon root, cation, anion uptake ratio and the form of nitrogen supply. So friends, the concentration of organic acid in the xylem sac depend upon the root, एक तो कितना cation, कितना anion, और दूसरा form of nitrogen supply. So this is also very clear that how the xylem transport are varied. Okay, now come to the next point, that is known as xylem loading. Okay, तो अब हम क्या कर रहे हैं, अपने न्यूट्रिएंट्स को जाइलम में लोड कर रहे हैं. तो यहां पर आपके क्या होंगे, देयर विल बी इन्वॉल्वमेंट ऑफ ट्रांसपोर्टर्स. ओके, सो द एलिमेंट्स that transverse the root via the symplastic pathway. You know that symplastic pathway is the major pathway of the solute transport. Okay, are loading into the xylem by various transporters proteins across the plasma membrane of xylem parenchyma. Okay, so there will be an involvement of transport protein before the transport into the plasma membrane of xylem parenchyma. So nitrate can be loaded in the xylem by a member of transporters NRT1 family. Okay, so NRT1 are important for the transport of nitrate into the xylem, and anion channels can facilitate the movement of nitrate, sulfate, phosphate and the chlorine. Okay, तो anion channels पे कौन कौन से आपके nitrate, okay, sulfate, phosphate, okay, then chlorine. Okay, so these are the major nitrate anion species and can be transported by the anion channels in the direction of their electrochemical gradient from the cytosol of the plasma xylem parenchyma cell to the xylem vessels. Okay, so movement by the difference in the electrochemical gradient means high to low. Okay, next point, तो ये क्लियर है नाइट्रेट आपका कैसे हुआ और एनायंस कैसे हुए. ओके, नेक्स्ट, बोरॉन इज लोडेड इनटू xylem by the Arabidopsis. AT means Arabidopsis thaliana BOR1 transporters. Okay, तो ATBOR1 involved in the boron transporters. Potassium loaded into the xylem by voltage gated outwardly rectified potassium channels on the plasma membrane of root pericycles and the stellar parenchyma cells Arabidopsis thaliana as core. Okay, So these transporters are involved for the potassium load. Okay, तो clear है. Next, cation that are present in low concentration in the cytosols of root cells are loaded in xylem by the active transport mechanism. तो बिल्कुल क्लियर है, जो भी चीज आपकी कम कंसंट्रेशन में प्रेजेंट होगी उसके लिए हमारा एक्टिव ट्रांसपोर्ट मैकेनिज्म की जरूरत पड़ेगी. ओके, सो कैल्शियम इज लोडेड इनटू द xylem by calcium ATPase and also the members of like heavy metals PiB1 ATPase load the zinc and the copper into the xylem. Similarly, magnesium and manganese are loaded by the ATPase, and cation carriers proteins also implicated in loading zinc and iron into the xylem. Okay, so some carrier proteins and some ATPase proteins are involved in transport of heavy metals like zinc, copper, iron. Okay, so this slide is very clear that how the nutrients are loaded into the xylem. So there will be a involvement of transporters proteins. It could be channels, carrier proteins, or the pump. Okay, next, a significant amount of calcium, zinc, iron, sodium, okay, they positive है सब, cations है, can also reach the xylem through on apoplastic route when these are present in high concentration. Major pathway आपका symplast है, but they also can be reached through the apoplastic pathway if there is concentration is high. Okay, so nitrogen is mostly present in xylem, it in ionic form, okay, which is ammonium or nitrate, although amino acids and amides also present. Okay, calcium, magnesium, zinc are likely to be transported in the xylem as cation or cation complex with organic acids. तो लास्ट स्लाइड में हमने क्या डिस्कस किया था, एक तो इनके लिए ट्रांसपोर्टर्स होते हैं. ओके, दूसरा क्या करते हैं, दे कैन बाइंड टू द ऑर्गेनिक एसिड्स एंड दे कैन ट्रांसपोर्ट. ओके, फॉर एग्जांपल, iron is transported mainly as a ferrous citrate. Okay, and a member of the multi-drug and toxin efflux that is known as MAT. Okay, ये आपका ट्रांसपोर्टर्स है और ये ABC ट्रांसपोर्टर्स है जो आपका मतलब कॉम्प्लेक्स कंपाउंड्स को ट्रांसपोर्ट करते हैं. ओके, इसके अलावा ATFRD3 is expressed in the root paracycles and appear to involve in loading citrate into the xylem. Okay, so these two transporters are involved. Okay, then zinc can also be transported as histidine complex and zinc, copper, manganese, nickel can be transported as nicotinamide. Okay, complex. तो ये क्या करेंगे, जिंक निकोटिनामाइड, कॉपर निकोटिनामाइड, तो ऐसे कॉम्प्लेक्स बना के इनके लिए ट्रांसपोर्ट करेंगे. ओके, ड्यूरिंग ट्रांसपोर्ट फ्रॉम रूट टू द लीव्स इन द नॉन लिविंग जाइलम वेसल्स, इंपॉर्टेंट इंटरेक्शन टेक प्लेस बिटवीन solutes and the both the cell wall of vessels. मीन्स जो आपका जाइलम में फ्लो हो रहा है. ओके, तो यहाँ पे आपका solutes और जो सेल वॉल्स है उसमें भी इंटरेक्शन होता है. Because in the last class we have discussed that in the cell wall there is a it is made up of pectin and there is a carboxylic group COO- which also involved in the cation exchange in the root wall and here the vessels wall. Okay, and the surrounding xylem parenchyma cells, the major interaction are exchange absorption of polyvalent cation in the cell wall as like root and uptake and release of elements, and because there will be also formation of apparent free space. Okay, and only solid by surrounding living cells. Okay, so xylem loading is very clear. First, if there is a concentration is less, there will be a active transport. If concentration is high, then there will be a channels. Okay, and also they can transport in the form of ions and the form of compound with the citrate or histidine or the nicotinamide complex, and some transporters are involved like MET, AT, FRD. And the next point is there will be interaction of xylem basal cell wall and the parenchymatic cell wall. So they are also involved in the cation exchange, and also they involve in the release of the element and earning solutes. Okay, and formation of this apparent free space in the xylem. So this is a xylem transport. Clear? Now come to the next slide. There will be a exchange, adsorption and xylem retrieval, uptake. Okay. So interaction between the cation and anion in the cell wall of xylem vessels are similar to those in the AFS of root cortex. So in the last class we have discussed about the AFS and the DFS, that is known as donor free space or the apoplasmic free space. So here also there is a formation of AFS in the xylem vessels. Okay, where the cation and anion exchange take place. Okay, so the long distance transport of the cation in the xylem can be compared with the ion movement in a cation exchange, and this cation exchange adsorption is not restricted to the xylem vessels. The cell wall of surrounding tissue also take place. Means not only xylem vessels, cell wall of the xylem vessels, but also surrounding tissue also take place in this exchange and the absorption of nutrients. Okay, so the degree of retardation of cation translocation depend upon the valency of the cations. Already discussed that high valency have high absorption and its concentration in activity, high concentration or high activity. The charge density of the negative groups, total negative ions present in the xylem, the diameter of the xylem vessels, and next is the pH of the xylem cell, the formation of the irritant. Okay, so the retrieved element can be stored transiently and permanently, like in the AFS of the root cortex, in the xylem parenchyma and other stem tissue, for transport from the xylem to phloem via specialized cells, or they can use in the xylem or they can transport to the phloem. Okay, so retrieval of the sodium from the xylem sac is therefore an effective mechanism of restrictive translocation to the leaf blade. So when the sodium concentration high in the xylem sac, they can be restricted in the leaf tissue by the transiently and permanently stored in the xylem parenchyma. Okay, so retrieval from the xylem sap in root and stem can be a determining factor in the distribution of micronutrients in the plant. Okay, so it will regulate the concentration in the upper tissue and the xylem. Okay, so like molybdenum has been accumulated in the xylem parenchyma of root and stem. Okay, so this is about the exchange, absorption and the xylem retrieval. It is like a root cortex, formation of AFS and the store of the transiently and permanently and ट्रांसपोर्ट टू द जाइलम एंड द फ्लोएम। तो बिल्कुल क्लियर हो गया कि जैसे आपके रूट में हुआ था, रूट में क्या हुआ था, वहां पे भी फ्री स्पेस बन गए थे, कैटायन एनायन वहां पर स्टोर हो रहे थे ट्रांजिएंटली और परमानेंटली। अगर आपको जरूरत है तो ये जाइलम सेप में मूव कर दिए जाएंगे और दे कैन बी रेस्ट्रिक्टेड एंड दे कैन बी ट्रांसफर टू द फ्लोएम। ओके, सो इट दिस आल्सो रेगुलेट द कंसंट्रेशन और ट्रांसलोकेशन इन द जाइलम। ओके, नेक्स्ट पॉइंट, release or the secretion. Okay, so the composition of xylem sap along the transport pathway can also change by the release or secretion of solutes from the surrounding cells. Okay, तो जो आपका xylem sap है, that concentration depend upon the First is the transport of the solutes from the root to the shoot. Okay. Second, the release or secretion of solutes from the surrounding cells. Okay. Example, in lawn legume supplied with the nitrate and nitrate concentration in xylem sap, as the path length increase, where the concentration of organic nitrogen glutamine is decreased. Where in nitrogen fixing legumes, the ratio of this organic amide to amino acid is shifted in favor of the amino acids, and the release of secretion of nutrients from the xylem parenchyma into xylem maintain continuous supply to the growing part of the shoots. So what happen in this legume crops? There will be an increase in amide to amino acids, and they can release or secrete nutrients when these amides and amino acids when needed. So there will be a continuous supply of the nitrogen in the nitrogen fixing legumes. Okay. In the period of ample supply to the root, nutrients are retrieved back from the xylem sap, whereas in the case of insufficient root supply, they are released into the xylem sap. Okay. Like magnesium is also often stored in the root cell and released to the xylem when the shoot become magnesium deficient. ओके, ये आपके एक तरह के बैंक का काम कर रही है कि जब आपके पास ज्यादा है तो वहां पर स्टोर कर दीजिए, और जब आपके पास कम है सो दे विल रिलीज ओवर द सीक्रेट. ओके, सो दिस इज वेरी क्लियर that how the concentration of xylem sap will depend upon these two factors. Okay, release and secretion and the retrievation. Okay, now next to the xylem loading. Okay, unloading. तो हमने सबसे पहले xylem लोड कर दिया था, अब xylem को अनलोड करते हैं एरियल पोर्शंस. So despite retrieval of the element along the pathway in the stem, most of the solutes and water are transported in the xylem basins into the leaves. Okay, तो आपका retrieval तो एक प्रोसेस है जिससे कहीं ना आपकी थोड़ी बहुत न्यूट्रिएंट सप्लाई हो सकती है. बट मेन पाथवे कौन सा है? जहां पे आपका xylem फ्लो आ रहा है. ओके, सो दिस इज द most of the solutes are transported in the xylem phloem. So xylem unloading is crucial. Water भी आपका साथ में आता है. Water is preferentially transported in the major veins to the side of rapid evaporation such as leaf margins or from the vein ending mainly by symplastic मूवमेंट टुवर्ड द स्टोमेटा. ओके, तो वाटर का आपको पता है कि वाटर का ट्रांसमिशन जहां आई होगा तो वाटर आपका मूव करता जाएगा कहां पे? लीफ मार्जिन्स पे. ओके, और वेन्स, लीफ वेन्स एंडिंग पे. Although the bundle sheath cells or bundle sheath walls of the veins are severized in leaves of C3 and C4 grasses, and they do not provide a barrier against the apoplasmic flux of water and solutes. तो जैसे आपने देखा कि in the root there will be a epidermis, there will be a some severized cell in the bundle sheath cell, but they are not barrier like as a endodermis in the root. Okay, they are less. So depending upon the concentration and composition of solutes in the xylem sap entering the leaf and the rate of water loss by the transferring along its stream through the leaves, the solute concentration may be enriched several fold at the leaf edges because this nutrient reach to the with the water flow. Okay, तो आपका वाटर है वो आपका लीफ में क्या होगा? वाटर फ्लो के साथ पहुंच जाएगा. तो लीफ एज पे आपका क्या होगा? न्यूट्रिएंट कंसंट्रेशन will be high. Okay, this is particularly true when element concentration is high in the root medium like boron and silicones. Okay, so they will be accumulate high at the leaf edges if some nutrients are transported in high concentration. Okay, unless some of this excessive solutes accumulation at the terminal site of the transferring stream is removed as a occurring for the boron or through the epidermal glands. So plants have some mechanism by which they can be secreted outsides or they can be effluxed outsides. Okay, like epidermal or salt gland in the salt gland in the halophytes. Okay, necrosis in the tips. If there will be a necrosis, this portion is removed and iron can be removed. Exercise and necrosis in tips and margin of leaves. Some plant accumulate high calciums, okay, in the leaf trichomes, that is leaf hairs, and that form the calcium oxalate crystal in specific cell types. They form the calcium oxalate. Okay, so prevention of excessive solute accumulation in the leaf apoplan by a mechanism other than uptake by the leaf cell can be achieved by the formation of salt of low solubility in the apoplan, or they form the salt of low solubility. Okay, means they form the complex. Okay, and a safe way of coping with a continuous xylem import of calcium by calcium oxalate crystal in gymnosperm, which cannot be exported in the phloem, means they can reach to the and they cannot retrieve back to the phloem. Okay, in a fast growth plant with low nutrient supply, the solute concentration in the xylem set decline sharply. Means एक प्लांट है वो जल्दी जल्दी आपका ग्रो कर रहा है, but there will be a low nutrient supply. So what happen in this case? Solute concentration in jar, तो आपका क्या होगा, आपका solute concentration है that is decline in the xylem cell from the root to the leaves or within a leaf blade from the base to the tips. Okay, the cells of bundle C cells are the site of intensive net proton exertion which acidify the apoplan. Okay, so in the bundle C cells there will be proton exertion or proton channels are active which acidifies the apoplan, and the electrochemical potential and proton gradient across the plasma membrane of leaf cells act as a driving force the solute uptake. तो जो आपका solute जालम से गया तो वहां पर बंडल सी सेल्स होती है, so there will be a high activity of the proton gradient channels and the formation of electrochemical gradients which transport the most of the nutrient to the these leaf cells. Okay, so nitrate retrieved from the xylem by the transporter nRT1 and nRT2, ammonium by AMT1, phosphate by the PST1, sulfur by the SULTR1 and 2, boric acid channels encoded by NIP, that is known as nodeline 26 intrinsic protein, aquaporins gene family. Okay, so they are for the xylem unloading in the leaf cells. So these transporters are involved in the xylem unloading. Okay, also the electrochemical gradient between the xylem shoot apoplast and the cytosol of the shoot cell suggested that the influx of potassium and calcium to the shoot cell can be mediated by cation channel in their plasma membrane. So friends, there will be involvement of several transporters which involve in the transport of this nutrient from the xylem cell to the leaf cells. Okay, so this is known as xylem unloading. Okay, तो वेरी क्लियर हो गया. तो पहले हमारा जाइलम लोडिंग हुआ, जाइलम लोडिंग के बाद जाइलम में सैप हुआ, वहां पर कुछ एब्जॉर्प्शन भी होता है, कुछ रिट्राइवल भी होता है, जाइलम फ्लोएम ट्रांसपोर्ट भी होता है, फिर वो जाइलम ट्रांसपोर्ट करके आपका एरियल पोर्शंस में आता है, एरियल पोर्शंस में आके उसका क्या होता है, जाइलम अनलोडिंग होता है, तो वहां पर भी आपके ट्रांसपोर्टर्स आर इन्वॉल्व. ओके, नेक्स्ट इज द फ्लोएम ट्रांसपोर्ट. ओके, तो जाइलम ट्रांसपोर्ट इज द मेजर पाथवे ऑफ द न्यूट्रिएंट पाथवे, बट दिस फ्लोएम ट्रांसपोर्ट आल्सो इन्वॉल्व इन द ट्रांसपोर्ट ऑफ न्यूट्रिएंट. ओके, मेजर तो आपका जाइलम है, but this is also involved in the nutrient transport. So long distance transport in the phloem take place in a living cell. This is known as sieve tube, already discussed. और xylem में अपन का कौन सा है, non living cell, that is known as xylem vessels. The principle of phloem transport mechanism was proposed in the 1930 by the Munch, which is known as pressure flow hypothesis, based on the principle of osmometer, means high concentration to the low pressure. Okay, high pressure to low pressure. Munch suggested that such as sucrose are concentrated in the phloem of leaves, that is known as phloem loading, and water is sucked into the phloem creating a positive pressure. ओके आपका वहां पर ये सुक्रोज भी है और वाटर भी है, तो वहां पर क्या हो जाएगा, हाई पॉजिटिव प्रेशर, and that caused by the removal of solids from the volume. Okay, therefore flow rate and direction of flow rate are closely related to the phloem unloading at the sinks. Okay, तो ये आपके source tissue है आपका leaf, that is known as source tissue. Okay, और सिंक यहाँ पर आपका formation हुआ and water sucrose, so this is high pressure and it's moved to the grain and another portions. Okay. For nutrient, the main site or source for the phloem loading are located in the stem and the leaves. So leaves and the stems are work as a source, and within the phloem, the sieve tube elements are associated with companion cells and the parenchymatic cells. They are involved in the transporting pathway, and a very large concentration of free calcium ions must be maintained in phloem sac for long distance transports. ओके तो यहां पर कौन कौन इन्वॉल्व होता है, एक तो आपका पॉजिटिव प्रेशर विच इज कॉज्ड बाय द सुक्रोज फॉर्मेशन एंड द वाटर, सेकंड आपका कौन सा ट्रांसपोर्ट्स के लिए कंपेनियन सेल्स, सीव ट्यूब्स एंड द पेरेन्काइमा सेल्स, एंड देयर विल बी सम फ्री कैल्शियम व्हिच आर फॉर द सिग्नलिंग एंड द लॉन्ग डिस्टेंस ट्रांसपोर्ट. ओके so this is phloem transport. Okay, now what is the difference between the xylem and phloem flow? Okay, so what is the difference? So in the phloem there will be organic solutes are the dominant solutes, जबकि xylem में क्या है आपका inorganic ions. Okay, like potassium, calcium. Okay, यहाँ पर क्या है sucrose. Okay, second transport take place in the living cells, कौन सा sieve tube cells. Okay, जबकि xylem में कौन सा xylem basal cells which is dead cells. Okay, third, the unloading of solutes at the sink play a important role. इसमें क्या है, this is depend upon the number of sinks and the sink activity. That's why it is known as source sink relationship. Okay, so these are the three difference between the xylem and phloem transport. Okay, next point is the phloem loading and its composition. Okay, अब this xylem shape का हमने कर लिया, अब phloem shape में आते हैं. So phloem shape भी है जे high pH. pH is near to 7 to 8. Also it contain a high concentration of solutes, on average 15 to 25% of dry matters. Okay, in which the main component is the sucrose. Okay, which are synthesized in the source tissue. Also some amino compounds like amide glutamine, asparagine, also citrate, malate, raffinose, stagose, sugar alcohols. So these are the important composition of the phloem sac. जबकि xylem sac में आपके कौन होते हैं, mainly inorganic ions like cations and the anions. Okay, next point is the mobility in the phloem. So macronutrients except calcium all can be moved. Okay, so mobility is high. Okay, micronutrients mainly intermediately movement except for the manganese. Molybdenum is fairly high, manganese is low, boron is intermediate and calcium is low. So this is the mobility in the phloem. Okay, next point, the transfer between the xylem and phloem. Okay, so in a vascular bundle phloem and xylem are separated by the only a few cells, and the exchange of solutes between the two conductive system is very important for the regulation of long distance transfer pathway. So friends, we already discussed that there will be an exchange of solutes between the xylem and phloem, and that is very crucial because there will be some barriers like root shoot junction, that nodes. So in this case there will be a less or there will be a blockage in the xylem. So in case that nutrient can be transported by the phloem, okay, and it is evident that transfer from phloem to xylem concentration occur down a concentration gradient according to their high to low concentration. In contrast, most organic and inorganic solutes, a transfer from xylem to phloem is usually against a steep concentration gradient. Okay, आपका phloem to xylem आएगा तो वो आपका होगा concentration gradient के आकार, means passive flow. Okay, आपको पता है in the phloem there will be the passive transport is dominant, but in case of xylem transport, active transport is dominated. Okay, so it will move against the concentration gradient. Xylem targets higher transmission site and phloem decrease demand sites. The stem nodes are the site of intensive xylem to phloem transfer, already discussed. Okay, now next point is the re-translocation or cycling of the nutrient. Okay, तो word क्या है? Re-translocation and the cycling of nutrients. So rapid xylem to phloem transfer in leaf blade, in which only a small fraction of total leaf content or cyclic fraction is involved. So this involved in the minute quantity. A considerable amount of nutrient are retranslocated in the phloem from the shoot back to the root and used to convey information about the feedback regulation control uptake by the root. So this regulates the feedback regulation that we have sufficient quantity of this nutrient. Okay, in plant, root provide the main site of nitrate reduction. Retranslocation of nitrogen in reduced form in the phloem from shoot to root is required to meet root demand for reduced nitrogen. So when there will be a reduced nitrogen, so that time there will be a retranslocation of nitrogen from shoot to the roots. And cycling is an important process for the maintenance of charge balance if there will be an imbalance in the ion homeostasis in the shoot and root. Okay, so in this case retranslocation and cycling of nutrients take place. And next point is, and last point is the remobilization of nutrient, and that is a very important process. So remobilization process is based on the range of different physiological and biochemical process. Okay, first, utilization of nutrient stored in the vacuoles, means how much nutrient you are already stored. Okay, second, breakdown of stored proteins or materials, and third is the breakdown of cell structure and enzyme. So these factors decide the process of remobilization or rate of remobilization. Remobilization of nutrients is important during the ontogenesis senescence of a plant at the following stage. So at three main stages like seed germination, period of insufficient supply to the root during vegetative phase when the growth is very high, and reproductive growth, and fourth is the in perennial period before the leaf growth. So in these four stages remobilization of nutrient is mainly take place. So we'll discuss first is the seed germination phase. So nutrients are remobilized within the tissue and translocated in xylem or phloem to developing roots and the shoots. Consequently, seedling can grow for at least several days without an external supply of nutrients. So friends, you know that in initial conditions, the germination phase, this is the seed, this is a early seedling, and this is root system. So in initial stage there is a less developed xylem. So they are, or xylem differentiation or development is not take place. So they are unable to transport the solutes. But for the growth, there will be a remobilization of stored reserve in the seed, and it can be moved to the your growing seedling. Okay. So first process. Second, at the vegetative phase. So during vegetative growth, the nutrient supplied to the root is either permanently insufficient if they are growing in the nutrient deficit soil, or temporarily interrupted when the demand is high. Okay, these two cases can be happen. And remobilization of nutrient from mature leaf to the area of new growth is thus the key importance for the completion of life cycle under adverse environmental condition. So friend, if there is a adverse environmental condition, stress conditions, or the nutrient deficient condition, so in this case some leaves or the mature leaves are senescent, and they will deprive their nutrient to the growing leaves for the completion of life cycle. Okay. Third is the reproductive phase. So at this growth stage, root activity and nutrient uptake decrease. Okay, because root activity is also decreased and nutrient uptake is also decreased, because root activity is also decreased because of the decrease in the carbohydrate supply, zinc competition. Because at reproductive stage there will be a increase in the number of zinc, that is also fruits or the formation of grains. Okay, so they are the important zinc. The remobilization of highly phloem mobile nutrients can lead to such a rapid decline in their concentration in the vegetative source because of senescence is induced and plant behave as self-destructive system, that is as PCD, programmed cell death. Okay, so remobilization is highly nutrient selective, and the corresponding discrimination against element which are either not essential or required only in very low concentration, means they can transfer only the important or the macro or micro nutrients. During the reproductive stage, the degree of remobilization of micronutrient and calcium is high compared to that during vegetative growth, and the extent of remobilization of micronutrients is closely related to the leaf senescence. Okay, so friends, there will be a 3 stage and 3 different mechanisms for the remobilization of solids. So this is a mechanism of the overall transport of the mineral nutrients from the soil to the xylem, xylem to the tissue, and also retranslocation and the remobilization process. So friend, I hope you understand with the concept that what is the actual concept of the transporting nutrients or solutes from the root to the shoots, also retranslocation process, remobilization process, retranslation of element from the xylem cells, how they can store formation of the apoplasmic free space, role of the different transporters in the xylem loading and the unloadings. So there are the different factors which regulate the mineral transport pathway. So thank you. If you have any problem, you can contact me. Thank you very much.